Good afternoon. Welcome to our presentation. Today we are presenting on developing fit-for-purpose external control arms to optimize submissions to regulatory and health technology assessment agencies. First, a public service announcement. I've been asked to remind you to please use headphones to listen to the presentation. But if you leave the theater, please leave your headphones on your seat when you exit. Thank you. So I'm Jen Wogen. I'm a Senior Director of Evidence Strategy at Genesis Research, where I've worked on a number of external control arm studies over the past several years, primarily in oncology. And this is my colleague, Craig Parzynski. He's a Senior Director and Head of Biostatistics and RWE Analytics at Genesis Research. And Craig has also led methodology on a number of external control arm studies. So our learning objectives today are to describe the framework for using real-world data to enable comparison, to enable a comparison group for a single-arm clinical trial. We are going to describe key regulatory and HTA agency concerns regarding methodological challenges to external control arm studies. We're also going to explore pathways to early engagement with these regulatory and HTA agencies when assessing the suitability of external control arm methods to complement a clinical trial program. So first, I'd like to address some definitions and talk about what we mean by an external control arm study. In an external control arm study, we are comparing clinical outcomes experienced by a clinical trial, a single-arm clinical trial, for a treatment of interest administered to according to a clinical trial protocol, to those outcomes experienced by a similar population external to that single-arm clinical trial. And these patients that we're comparing to the single-arm clinical trial in the external control arm can be either treated or untreated. The key really is that the external control arm should be as similar as possible to the single-arm trial population to ensure, and to do that, we want to ensure that the data that we're choosing is really fit for purpose. At Genesis Research, we've worked with a number of external data sources to create the external control arm for comparison. These include historical trial data, electronic health record and claims data, as well as registry data. So we can create a trial-eligible population by essentially mimicking the inclusion and exclusion criteria that we have within our single-arm clinical trial to our external data source. We can also then apply statistical methods to create our target population, which is receiving standard of care or a physician's treatment choice, and then this becomes our external control arm. So when are ECAs used? Well, they're often used to contextualize single-arm trial data, and this can be by way of either a formal efficacy comparison or for benchmarking purposes. They're often used when it's infeasible or not ethical to use a placebo comparator, when the standard of care may be very likely to be updated during the course of the single-arm clinical trial, and often they're used in rare disease and oncology therapeutic areas where there may be issues related to adequate numbers of patients recruited in the single-arm in the trial. So the FDA recently released draft guidance for the conduct of external control arm studies. And this was important because the release of this draft guidance document earlier this year provided some key insights into the agency's priorities and expectations for external control arm studies. So while guidance from EMA and European Health Technology Assessment Agencies is applicable as well, for the purposes of a lot of this discussion, we'll focus on the FDA. So the FDA's guidance focused on the design and analysis of externally controlled trials using patient-level data from other data sources, such as clinical trials or from real-world data. But the key is really that the guidance focuses on patient-level data to use as an external comparator. Overall, the guidance stresses the need for early and well-considered planning of your external control arm study. The guidance recommends incorporating external control arms as part of the overall development program for the clinical trial. And the guidance recommends that straightforward approaches are generally preferred to complex ones with many assumptions. FDA recommends that the optimal real-world data source be chosen by a series of feasibility assessments. So we want to assess potential data sources to ensure that they are fit for purpose, and that we can adequately apply the inclusion and exclusion criteria from the single-arm clinical trial. We want to make sure that we have comparable endpoints that we are able to assess. We also want to ensure that we are able to measure similar prognostic variables. And then we also would like to be able to understand the extent of missingness in the proposed external data source. And all of these things are key to really assessing whether the data is fit for purpose for our external control arm study. Our goal is to use target trial emulation methods to ensure comparability between the trial and the external control arms. And some considerations are listed here at the bottom of the slide. So I'd like to talk a little bit about why early design is so important in the ECA process. If these design issues are not addressed adequately in the early planning phase, under review of the evidence generated by ECAs, by HTA and regulatory bodies, these issues can resurface as methodological criticisms that can potentially compromise the use and interpretation of the evidence generated from the external control arm. I'm on the slide showing an example of a recent review paper by JAXA and colleagues. There have been a number of recent review papers that have summarized regulatory and HTA agency criticisms and feedback of ECAs included in evidence submission packages. This particular example is by JAXA and colleagues, and JAXA and colleagues focused on 7 case studies in oncology that were reviewed by 3 regulatory and 5 HTA bodies. And this review is particularly informative because they assessed the types of critiques that were given by regulatory and HTA agencies, the number of criticisms or the level of criticisms that each of these studies received, but they also assessed the level of influence that the ECA evidence ultimately had on the regulatory or the HTA agency's ultimate decision. So one of the key takeaways here is that there's a lot of inconsistency between HTA and regulatory bodies, between specific regulatory and HTA bodies, related to specific methodological concerns identified within the same submissions. If you look at the example on the bottom row of the table at the top, this was a submission in relapsed refractory multiple myeloma. It was reviewed by 2 regulatory agencies, EMA and FDA. It received a fairly large number of criticisms across these 2 regulatory bodies. and was ultimately deemed to have a high influence on the EMA's decision, but the FDA considered that there were methodological concerns that were too significant to overcome, and the submission did not have a high influence on the FDA's ultimate decision. So this really stresses the need that feedback from regulatory and HTA agencies can be quite varied, even for the same specific submission. And although there are efforts underway to sync criteria across these regulatory and HTA agencies, it really stresses the need to engage these agencies early in the design process, so that the design of the ECA, early in the design of the ECA, any concerns related to data and methodology can be assessed in the design phase. And it really makes a point for engaging these agencies early in the process. Now, the specific issues that were most frequently identified across these case studies were concerns related to selection bias and unmeasured confounding. But FDA also, in their guidance document, stressed the importance of consistency of outcome definitions. In the next few slides, my colleague Craig is going to address each of these methodological considerations in turn.
Thanks, Jen. My name is Craig Parzynski. I'm a statistician by training, and what I'll go ahead and do is focus on each of the key concerns that Jen highlighted, and hopefully what you'll take away from this is that statistics can't fix bad design. And so we'll focus a lot on how we think about fit-for-purpose data to address these concerns. The first concern that Jen mentioned is selection bias, and in particular, this is when characteristics of trial patients differ from the real-world patients due to the selection process itself. We can try to mitigate this by applying all of the eligibility criteria from the trial to our real-world data, but of course this is only going to mitigate the concerns. One recent ECA that was reviewed by FDA in the metastatic urothelial cancer, selection bias was a key concern that was raised. And in particular, they highlighted that those in the trial were treated at academic centers, while those in the real-world data were treated in community centers, that the majority of the trial patients were from European countries, whereas the real-world data source was entirely US, and that many of the eligibility criteria were not able to be applied in the real-world data set. At Genesis Research, we've conducted several of these external control arms, and some additional considerations that have come up through our practice is one, related to the selection of the index date, and in particular, because we're dealing with retrospective data, patients who are eligible for our external control arm can be eligible at multiple time points. Choosing the correct time point is an important consideration because we can introduce selection bias if we choose the wrong one. And we've done a bit of work on this that shows either selecting the first or all of the eligible lines will be the correct selection, but something like the last line would be introducing selection bias. Similar to that is different study entry processes. So of course in trials, there's a screening phase in which all the assessments to decide whether you can enter the trial are conducted. But in the real world, the time at diagnosis and until the time of treatment might be quite different, and we need to think about that, in particular, something called immortal time. Similar to that is the timing of those assessments in the real world. And so in the real world, those assessments can happen near to the start of treatment or further away. And again, whether or not that means that they're actually eligible to enter the trial, again, we might have some selection bias that enters. One thing that happens that is quite often in these external control arms is that we end up using non-concurrent real-world data. And so ideally, we'd like to use data that coincides with the conduct of the trial. However, in practice, due to sample size concerns, we often need to use non-concurrent controls. And what happens in that scenario is, again, we might have things like changing standard of care that introduces a different type of patient into our eligibility for our trials. The last two bullets on this slide here are similar to what I mentioned in the FDA review, but are key concerns in the fact that it's rare that we can actually apply all eligibility criteria, and that geographic variability can play a concern in selection bias. Next, I'll talk about confounding. And in particular, we know that randomized controlled trials are the gold standard because, due to the randomization process, both observed and unobserved confounders can be balanced. In the real world, of course, treatment results from several different factors, including practitioners and observed patient characteristics, which lead to differences between our trial patients and those that can be enrolled in the real world. Resulting from this is confounding, and we end up with a biased, naive treatment effect. This was pointed out in an FDA review of acute lymphoblastic leukemia, where several potential confounders were not adjusted for in their analyses, and using statistical methods, they still had unbalanced prognostic factors. And in fact, many of the patients were removed due to the analyses that they conducted. So it brings up questions of whether or not they're in the same target population. Lastly, they pointed out, due to changing standard of care, that patients receiving stem cell transplant were quite different in the trial compared to the real world data. We experienced similar things in our studies that we've conducted, but a couple additional considerations are missing data in particular, and how to manage that and what that does for the differences in individuals that are able to enter the trial in the real world versus those that were captured, because missingness in real world data is much more prominent. Unmeasured prognostic variables, which again speaks to fit for purpose data, the statistical methods that are available to manage confounding do require that all measured confounders are available in the data set, so we need to identify data sources that are relevant and include all of those important prognostic characteristics. Inconsistent study definitions, and this happens quite a bit in the trial. Many of the important variables that are collected are defined in specific ways, whereas the granularity in the real world data may not be available. And how do we manage that from a confounding perspective when we need to remove the impact of those prognostic variables? And then lastly, the influence of temporality on standard of care. So I mentioned this in selection bias, but they highlighted it, the FDA, as part of confounding, and that we can't achieve balance between something like stem cell transplant. The last concern I'll talk about is the inconsistency in outcome definitions between trial and real-world data sets. The FDA particularly highlights this in their draft guidance, stating that there should be a focus on misspecification of the outcome and what that means for the validity of your external control arm. So for us, what we need to do in the design phase, when we're thinking through, as Jen mentioned, the target trial emulation, what the comparability of those endpoints are, their measurement, their reliability, and their validity. How similar are they, and does this create some fatal flaws in what we're trying to do? In our practice, in most cases, real-world endpoints are not fully aligned with the trial. And so how do we manage that through design? Do we look for another data source? Do we try to manage it through changing definitions? We need to consider these in the design phase. The FDA pointed out outcome concerns in an ECA on non-small cell lung cancer, where they pointed out that time to treatment discontinuation is complicated by treatment beyond the disease progression, and that progression-free survival is limited by missing or infrequent radiographic imaging in our real-world population. Some of this can be addressed through sensitivity analysis, and we've done that sort of analyses in our experience. But this does continue to persist. So in the real world, and from our experience at Genesis Research, incomplete or misspecified endpoint information is one of the biggest hurdles. For example, survival in real world data sets is often curated through additional external resources or sources such as obituaries or the Social Security Index. And in reality, it's very unlikely that all deaths are captured, and whether or not those are systematically captured or if there's bias introduced, those are things that we need to consider. Similar to that, in the real world, we are dealing with undefined criteria as to what progression is, for example. And so what that leads to is variability and subjectivity in the reporting of that outcome. again, another potential source of bias when thinking about the outcome definitions. Of course, in trials, we're using things like RESIST and Lugano criteria to define our endpoints. And what does it mean when those are misaligned? And how do we circumvent those? And can we circumvent those? And then lastly, which is what I mentioned previously already, is the unsystematic timing and frequency of endpoint assessments. And this does come up quite a bit, and in some instances have been fatal flaws in some of the studies that we've tried to conduct. I'll say the recommendation was not to conduct them in those scenarios, but there are things that you need to think about when we're designing real-world external control arm studies with trial. So I will pass it back to Jen now to wrap us up with some key considerations for engaging regulatory bodies.
Thanks, Craig. So given what we've discussed here today, it makes a very strong case for engaging regulatory and HTA bodies early in the design phase. Engagement, I think, ideally should start during the design phase prior to selection of a data source for the external control arm, to ensure that the data is really fit for purpose, and that the regulatory or HTA body of interest, or that you're targeting, is really in alignment that data source is fit for purpose. So ideally, engagement should occur prior to some of those decisions being finalized, so that there's still the ability to make changes to things like the source of the data for the external control arm and the methods that are being proposed to mitigate potential sources of bias. We strongly recommend engaging with regulatory and HTA agencies prior to the conduct of the single-arm trial, and ideally, FDA recommends that they would like to see a protocol before the start of the single-arm clinical trial. It's important for sponsors to assess the optimal timing of engagement, and also to assess what information is needed to take to these agencies during early engagement, what questions sponsors have for these agencies, and careful planning and provision of a protocol and a statistical analysis plan is recommended during these early consultations. It's important for us to mention, though, that such early engagement, although we really recommend it, it's not necessarily a guarantee of acceptance, but it can help potentially foster a collaborative experience with these agencies, and get their input early, before it's too late in the process to make changes to key decisions and to key methods as part of your external control arm. For EMA, non-binding scientific advice is available at any stage, and protocol assistance is available for orphan medications. For HTA in Europe, joint scientific consultations are recommended after feasibility assessment and proof of concept, but prior to study kickoff. And I'm going to end with a note that is another example or a cautionary tale about a recent submission, again, a different submission in relapsed refractory multiple myeloma. In this submission to the FDA, the sponsor did not submit the protocol before the conduct of the external control arm study, and the sponsor was only made aware of it at the results phase, at the presentation of the results phase. And the FDA ultimately ended up stating that they could not be sure that the protocol and the methods were pre-specified because they were not afforded the opportunity to review the protocol prior to the conduct of the external control arm study. So to summarize, I'd like to just stress a couple of key points. External control arm studies require early planning and thoughtful consideration and design to mitigate potential sources of bias and really maximize the value and the utility of the evidence generated from the external control arm study. We've made our case by highlighting some regulatory and HTA agency common criticisms related to methodological challenges often encountered in external control arm studies. And these key methodological concerns that we highlighted today include selection bias, unmeasured confounding, and non-comparability of study endpoints. And then finally, just to summarize, our recommendation for early and collaborative engagement with regulatory and HTA bodies to align on study objectives and key methodological components of the study, including selection of fit-for-purpose data and appropriate statistical methods that are pre-specified. Thank you very much. We'll take questions now, and if you have any additional questions, I think we have about 4 minutes, and would be happy to take any additional questions at the Genesis Research booth.
I have a question for you. I'm curious if there's been any precedent for doing sort of a bridging study or previous analysis to demonstrate adequate population comparison between the external population and the study population.
So, I think it's been proposed. I think the methods that exist currently, the way that we typically approach this is to address the data first, and focusing first on seeing what your trial looks like, and then identifying a data source that contains all the key data elements that you need to be able to actually identify your target population, to me really should be the first step. I mean, could you do that? Yes, I think you could. I haven't seen any examples myself. And so once you get through the exercise of identifying, a fit-for-purpose data set, you can use empirical evidence through statistical methods to say, if you look at these two populations side by side, I think we have it right. But that doesn't require the thoughtful design up front to think about, okay, what is our appropriate standard of care? Can we find that group? Can we manage them? And can we make sure that we're getting them correctly in the data itself? So I would focus more on getting the correct data source first. So one thing I'll add, the thing that happens most, I think, when I'm giving these types of talks is just questions about what types of feedback we've received. And most of it boils down to design phase issues. So hopefully that's what you've taken away from this, in that we most frequently hear concerns about comparability, either of populations, the target population, or comparability of endpoints. And so if we can get to a place where the data can support identifying the correct target population, and then ensuring that we have objective outcomes measured in that real-world data set, I think we'll be in a lot better place for using these in regulatory situations where, in most of our instances, they've been used as support but not actually as evidence. And so I think there's a little bit to go still in terms of data sources to be able to actually address and use these in regulatory submissions. So much work to be done. Thank you, everybody.
Thanks, everyone.